Alrighty, welcome to 26-5 and 26-6. Here we'll just add a little bit more to the story of what's happening at the renal tubule. We already gave a pretty good description with our picture that we made earlier, so you can use that to reference as we talk about just some vocabulary that we need to mention about different transporters and such. So, renal tubule, like we mentioned, functions to reabsorb all useful organic nutrients in the filtrate. So, as urine is going through the PCT, nephron loop, DCT, and collecting duct, we're trying to reabsorb some of the good stuff in there. We also work to reabsorb water. So, the majority of the water gets reabsorbed in the collecting duct and then in the descending limb of the nephron loop. Secreting into the tubule lumen, we're going to secrete in there any wastes that did not already get filtered at the glomerulus. We just as a reminder, reabsorption means that we're moving things from the tubular fluid through the simple squamous or the cuboidal epithelium of the renal tubule, and then back into the into the interstitial space, and then from the interstitial space into the bloodstream directly. Tubular secretion adds substances to the tubular fluid. As we gave some examples, sometimes we're dumping in things like hydrogen protons and potassium ions. Okay, so this is a little bit of a recall, but we need to understand the different types of transporters that we might have. So with facilitated diffusion, we're still following a concentration gradient, and we don't need any ATP, but it's facilitated, so we need to have some sort of help. It's not like water, it's not like osmosis, where we can just move from a high to low concentration and things just flow in between the cells of the membrane. But instead, we actually have to have some sort of help in order to bypass that membrane. So that's facilitated diffusion. And then we have active transport. Active transport, it's active because we need to use ATP. And this is where we're exchanging and pumping against or with the concentration gradient. So this is typically what we talk about whenever we're talking about neurons, wherever you're using a sodium, potassium, ATPase, sodium, potassium, ATPase transporter, because you're moving sodium against the concentration gradient, so you're pumping sodium into an environment that's already positive, or that has more sodium ions. And so this, you can imagine if we had a cell like this, and we had the co-transporter here, this would be activated by ATP, so you could imagine it kind of like spinning around, and as it's spinning around, maybe it's bringing sodium in and exchanging for potassium out. But this is considered active because you actually need to input energy into that transporter in order to move those ions across that membrane. Co-transport, this would be almost the exact same thing, except you don't need to use ATP. So we're co-transporting things, and two things are moving into the space. So this would be like moving two sodium and chloride into the cell, and they're co-transported, so both of them are moving in the same direction, and we don't necessarily need to have any ATP involved here, but we might have some carrier proteins helping bring this in. So that could be like this integral membrane protein here. We're also potentially we're actually following the concentration gradient for co-transport. For counter-transport, that would be in the total opposite direction. So if we drew the same thing here, but instead now we're moving chloride and sodium out of the cell, that would be counter-transport for two ions. And a membrane can have many of these multiple carrier proteins. So even though the membrane is really, really small and cells are really, really small and it's hard to kind of visualize what's going on, you really have to picture the plasma membrane as this really, like this horizon, this never-ending horizon of all sorts of things that are going in and out of the membrane, that are floating on top of or just beneath the membrane or floating through the membrane, and that are holding the membrane together. There's a lot of activity at the membrane. So you can't forget that we might have things literally right next to each other. We might have things on opposite sides of the cell that are doing the exact same thing, but we might have tens of thousands of them, but we might have lots of them, not just one. So you're not only just moving one sodium ion, you're moving one times however many of those co-transporters or whatever across the membrane. The apical aspect of the tubular cell may have different carrier proteins than the basolateral side. So whatever happens at the apical membrane may not be the same as the basolateral side. This is a really good example of what we talked about during the digestive chapter, where we were talking about how the parietal cells pump out hydrochloric acid and how do they do that. lots of activity was happening in those parietal cells. We were using carbonic anhydrase to change or to convert CO2 and water into H2CO3, and then from H2CO3 into hydrogen protons and bicarbonate ions. But what we saw was that the hydrogen protons were actually leaving on the opposite side of the bicarbonate ions. And in order for those could have happened on the apical side of the cell, but what actually came in on the basolateral side of the cell was CO2 and water. So things like this are happening all the time also, which makes it kind of difficult to keep track of whenever we're talking about a large system. So that's why we like to break it down by parts of the nephron and by what ions are being exchanged back and forth. We also saw another example of this in the collecting duct, where we had the aquaporins get activated on the basolateral side and then they moved to the apical side, and it was only at the apical side that we could actually move water in. And then the basolateral side allowed for diffusion of water out. That's another example. I can't think of any other examples off the top of my head, but we this is more of a conceptual piece. We wouldn't ask you like to provide a specific example. Two things about the renal tubule that are specific to the renal tubule are transport maximum and renal threshold. So transport maximum is abbreviated T subscript M, and it can be thought of the number of carrier proteins that are available at the plasma membrane of a cell. And the number of carrier proteins available at the plasma membrane are essentially the threshold of how much stuff can actually get even passed through that plasma membrane. So if the transport maximum is fully saturated, that means we have a bunch of carrier proteins at the plasma membrane, and it's full. There's no room for anything else. And you further increase the substrate concentration. Oh, and I'm sorry, I should add one other thing. When they're fully saturated, they're also being used at that time. So we're exchanging ions at all of those carrier proteins, and we only have as many carrier proteins as the plasma membrane can even hold. So what happens if that's the case? We're fully saturated, and then you further increase the substrate concentration. What would be the effect of the rate of the reaction? Well, the effect of the rate of reaction would stay the same, because the X amount of carrier proteins you have is the rate at which you're going to be able to transport those proteins or ions. And if they're all fully saturated and you further increase the substrate concentration, you're not going to be able to undo the fully saturatedness. You still have to wait for all of those carrier proteins to move those ions across. So that increase in substrate concentration is just going to pass through and not get reabsorbed like it normally should. So you'll end up losing ions in the filtrate as opposed to reabsorbing them. So having it fully saturated is not necessarily a good thing unless you actually have like a normal amount of filtrate. But whenever we talk about transport maximum actually being fully saturated, it usually doesn't mean that good things are happening. A great example of this is with diabetes. So if we have a lot of leftover glucose in the bloodstream, and then that blood gets to the kidneys, and then the glomerulus filters out the extra glucose monomers, and then those, monosaccharides, excuse me, and then those go into the proximal convoluted tubule, and that's where we absorb most of the glucose. Well, if we only have, let's say, 5 carrier proteins at the PCT, and that's the max amount of carrier proteins we can have at the PCT, then only 5 glucose molecules are going to be able to be absorbed at any given time. But if you have a flood of glucose passing there, and only 5 out of the 15 molecules that are passing by get reabsorbed, the leftover 10 are just going to continue into the filtrate, missing their chance to be reabsorbed, and they'll end up in the urine, which is why diabetes is called diabetes mellitus, because that means sweet pea. So that's where we actually get that overflow of glucose into the urine, or it spills into the urine is usually how it's said in the clinic. And then transport maximum determines renal threshold. So this is this is exactly what I was talking about with the glucose. The renal threshold is how good is the nephron at actually collecting the ions and reabsorbing the ions. And the renal threshold, there's a normal renal threshold, and if we have more ions than the renal threshold can reabsorb, then the nephron is failing. Table 26-3, I thought this was a really useful table to kind of independently talk about what things are being reabsorbed and what things are being secreted and what things are going to end up in the filtrate no matter what, because we have no way of transporting it across the membrane of the nephron. And this is just like a good starting point if you're still confused on what is in and out of, or what is and what isn't urine and what is or isn't filtrate. This might be a good starting point for you, so I included it in the notes. So now we'll talk about the PCT, the nephron loop, and the DCT, but a lot of this we've already mentioned, so this is a little bit more of a review. We already talked a lot about this in 26-3 or 4, where we were actually showing what's happening in the nephron tubule. So in the PCT, the majority of absorption happens here, 60 to 70%. Then once it's reabsorbed, all of that extra glucose, sodium, chloride goes into the peritubular fluid and then into the peritubular capillaries, as we drew previously. Here we're reabsorbing glucose, amino acids, and sodium and potassium and bicarb, magnesium, phosphate, sulfate, water, and ions. That's a lot. The biggest ones that we really care about here are glucose and sodium and potassium and bicarb. The other things are really small, and even their passive reabsorption, as you can see on their list here. The ones that are actively reabsorbed, that's what I said, that means the ATP is involved, and glucose amino acids, those are either facilitated across the membrane or they're co-transported across the membrane. So glucose usually gets co-transported with sodium. Amino acids usually get co-transported with sugars. So there could be transporters that are shuttling amino acids and glucose as well. Ions, this is active reabsorption. So they are passing along, going from a high to a low concentration, going against the concentration gradient, but with ATP helping it. And then passive reabsorption, we're just going from a high to a low concentration. Secretion in the PCT doesn't really happen here except for hydrogen protons, and that's the only one that we mentioned in our diagram as well. Your book does a good job of kind of showing you cell by cell what's happening in the PCT versus DCT versus nephron loop. So here's one of those images. If this visual helps you a lot more than the previous diagram we drew, by all means, please use it. What it's showing you is the tubular fluid up at the top that's in yellow, and then the peritubular capillary down below, so it's just inverted from the image that we had drawn. And then the PCT cuboidal cell in the middle with lots of microvilli there, so we know we have lots of reabsorption happening here. It shows you down below in the key, it shows you which ones are being counter-transported or co-transported. So like sodium moving in with glucose versus potassium and sodium being exchanged. And it also shows you like the osmosis of water flow. So we're going from a low to a high from a high to a low concentration. Excuse me. What else? I believe that's really it. So this might be a really good tool for you to learn it. But if the previous diagram works, like I said, please use that. All right, moving on to the nephron loop. This is where the descending limb we're reabsorbing water, and then the ascending limb we're not reabsorbing water. We're not permeable to water at all. We're only moving out solutes, so we're reabsorbing those solutes. And like I said, both the descending and ascending have thick and thin portions. Your book stresses that the ascending limb is the only one that has thick and thin portions. We don't go into the specific details of what's happening at the thick or the thin. It's just more important to understand that we're reabsorbing solutes, and mainly sodium and chloride here. The solute concentration is also increasing to concentrate urine. So remember, at the bottom of the nephron loop, the closest to the medulla, we have the highest solute concentration. So we have the least amount of water there and the most amount of solutes. Then we move to the distal convoluted tubule. This is where that fine-tuning happens. We're selectively reabsorbing things like sodium and calcium if we haven't already taken care of it. We're selectively reabsorbing water in case the nephron loop didn't do a good job, but we know the collecting duct is going to do most of that. We might secrete in ions, acids, drugs, and toxins. So if the glomerulus already filtered it out, but maybe it missed a couple, now the peritubular capillary is also going to dump it into the tubular space. We talked about how aldosterone and other hormones affect the distal convoluted tubule. Aldosterone specifically is pumping in hydrogen protons, which actively promotes reabsorbing sodium ions. And when we're reabsorbing the sodium ions, that means we're exchanging them for potassium ions. And so we're losing potassium ions. They're coming out of the blood and going into the tubular space, going into the urine. And so if aldosterone's turned on for a really long period of time, we can end up with something called hypokalemia or low potassium levels. This can actually turn into alkalosis, which means a really basic environment of the bloodstream, which just makes sense because we're also stimulating protons to be dumped into the tubular space, so we're losing the ability to have we're losing the acid that would buffer out the base that's left over in the blood. We also talked about parathyroid hormone and how parathyroid hormone is promoting calcium reabsorption here. I always think of parathyroid hormone. I always know it's antagonistic to calcitonin, and I remember calcitonin is bone-in, so we're taking out calcium from the bloodstream and putting it into the bone, so we're decreasing the amount of calcium that's available in the blood. Versus parathyroid hormone is going to do the opposite. We want to get more calcium in the bloodstream, so therefore we need to pull it out of the tubular fluid. We also talked about how NH4 is the one that's getting secreted here into this space, and that's really important because ammonia is the main molecule that really gives urine the kind of smell that we're familiar with. I don't even want to describe it. You all know what urine smells like. Again, here is a picture that your book gives you with the tubular fluid being at the top and the peritubular capillary being at the bottom, and the exchange. If this is helpful for you, awesome. The picture A is normal, B is talking about how the normal, and then how aldosterone can promote those co-transporters specifically. No, countertransport, countertransports exclusively. And then C is what's happening all together. Now we need to mention something and then finally we have the collecting system, which is at the end, the collecting duct. We talked about how aldosterone also affects the collecting duct and promoting sodium reabsorption, which again is pumping more potassium into the filtrate, or into the pre-urine. We talked about how bicarb is being reabsorbed for that acid-base buffer. And we talked about how the aquaporins are turning on and promoting reabsorption of water. Now we need to address something called the countercurrent multiplication, or the countercurrent exchange. It's been called it's called both of those things. So if you're Googling and you're trying to find good graphics, Google both of those terms. And this has to do with remembering that the solute concentration is really, really high at the nephron loop, at the bottom of the nephron loop. And the whole point of countercurrent multiplication, before we talk about how it works, is it is one of the best mechanisms we have to control the amount of urine volume we have. So remember, the descending limb is reabsorbing water, and so by the time we end up at the bottom of the nephron loop, we have a really, really high solute concentration. And we really don't have a lot of water left over, but if we do, the collecting duct will take care of it for us. So we're trying to concentrate urine as much as possible by the time we get down here. And we need to remember another thing. We drew the peritubular capillary around the entire nephron, but we need to remember that at the beginning we talked about two different types of nephrons. We talked about cortical nephrons and we talked about juxtamedullary nephrons. And we talked about how cortical nephrons are at the cortex, and they dip a little bit into the medulla, but not a lot. Those are the ones that have mainly peritubular capillaries. They don't have anything else at the nephron loop. Versus the juxtamedullary nephrons, those are the ones that have a vasa recta, and that's the specialized blood supply in the nephron loop. And now we need to talk about why that is and why it even has a specialized name. So the juxtamedullary nephrons, those are going to be deeper in the medulla, and if we're deeper in the medulla, that means we have an even higher solute concentration that's surrounding us. So the medulla is a really, hypertonic space. There's lots of solutes in that area because we have a lot of transport of solutes happening in that area. And in order to make sure that we get the most amount of reabsorption happening in this area, we need the vasa recta, which let me draw in red here. So if we're talking about a juxtamedullary nephron, so with vasa recta. And that vasa recta is going to be specific to the nephron loop. And the juxtamedullary nephrons, tend to sit kind of like this, where the cortex is up here and the medulla is down here. I'll just do capital M down here. And the medulla is also going to have a really high solute concentration because we have lots of this exchange happening back and forth. So this interstitial space, we get a little bit of ions there because we didn't do such a good job of exchanging them. And so we have to account for this. And in order to account for this, the vasa recta is going to be moving in the opposite direction of flow of the nephron. So what does that mean? So imagine that the nephron is moving in this direction, right? Filtrate is moving down in this direction. Da-doom, 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 da-doom. That's what we've already learned, right? The vasa recta is going to be moving in the opposite direction. So the arrows are going to be pointing this way. So we can start with the vasa recta up on this side. And the vasa recta, as it comes down here, slowly collects more and more solutes from over here, right? Because we continuously absorb sodium and calcium, or sodium and chloride. So sodium and chloride keep dumping in, keep dumping in, keep dumping in, right? And so by the time we get here, we're also going to have a really high solute concentration. So really high solute concentration in the blood and a really high solute concentration in the filtrate. Okay. As we keep moving up back towards the cortex, we want to dilute that solute concentration. So as we gain more of those solutes, now we want to dilute it, and because of that, water is going to want to move out of the nephron from the descending limb and out into the vasa recta. So we keep doing this in order to dilute the solute concentration, and by the time we get up here, hopefully we're at about equal solutes to water, or we're in an isotonic solution. This is why the descending limb is also specifically only permeable to water. It will only let water go in and out, out, so that we can help dilute this. So we can help dilute this vasa recta as much as possible. We also need to think about just this section for a second too, and why would we move solutes into the bloodstream, especially potassium especially sodium and chloride ions. Doesn't that seem a little strange to pump so much into one area of a blood vessel? Well, you have to remember that these are really, really small, and they're part of a huge network of capillaries that are everywhere. So this is just one small vessel in a big, big complex. And the reason why more and more ions move into this space, right at here, we probably don't have as many ions coming in versus down here we have a lot of ions coming in. And so in order to promote more of those ions to get into this space, we need to have a really high solute concentration here as well. And this makes sense too, because here we're going to have a really high solute concentration greater than this side of it. So because we have a higher concentration than over here, we'll move from a high, this is also a high solute concentration, so we're going to have to actively pump against this, which is why we have active protein channel, active co-transporters or counter-transporters here in order to actively pump sodium and chloride into the vasa recta, with the whole goal of hopefully getting diluted on the opposite side. All right, so this whole concept is called countercurrent multiplication. Multiplication in the sense that you keep concentrating the filtrate as much as possible and reabsorbing as much as possible. I don't love the term multiplication because I think that seems kind of confusing, but it is an exchange, right? On one side of the vasa recta, you've got a lot of ions being exchanged. On the other side, you have a lot of water being exchanged. So whichever term makes more sense to you, stick with it. If you want to remember both, great. This picture also gives you step-by-step what's going on from what I talked about here in this picture. Okay. We already talked about how aldosterone works on the distal convoluted tubule and the collecting duct. 26-6 also mentions this as well, where ADH works on both the DCT and the collecting duct in order to promote water reabsorption by increasing sodium reabsorption. Thus, we bring in more water so that we increase blood pressure. Here's a nice picture of how ADH is working on the DCT and on the collecting duct. Same thing we already drew before, but again, if these images make more sense to you, by all means go for it.